Hello, everybody. Welcome. This is lesson 5 in our muscular system unit. So we're going to shift gears a little bit today, and so hopefully you'll understand where we're going. We talked a little bit about how the muscle moves, but we're sort of backtracking here and saying, okay, where do we start this? And of course, uh, the vast majority of signals going to your muscles come directly from your brain. There are a few exceptions, but we'll not get into those right now. But for the most part, we need to talk about how does that message get to the muscle. So we're going to sort of backtrack and we're going to work our way back towards the muscles. So the nerves that actually attach themselves to muscles are called motor neurons. And their job is to essentially tell muscles when to contract. There is no signal for a muscle to relax. The brain doesn't have to send that signal. It just stops sending the contraction signal. So that's part of the concept here. And so we start off in the central nervous system. The motor neurons do not necessarily run directly from your brain. In fact, most of them run from somewhere in your spine. So there are places in your spine where they start, but they are connected to your central nervous system. And essentially, the original signal comes from your brain. So nerves are one way. That is one common misconception. So, there's that old idea of you put your hand on a hot stove and your brain senses that it's hot and then it pulls it away. And people mistakenly assume, okay, there's a signal going to the brain and then a signal coming out from the brain. They're both the same neuron. No, that's not the way it works. Every neuron is a one-way street. So we have sensory neurons in your hand that can actually sense things like heat. They're going from the outside back to your central nervous system, whereas these motor neurons are coming from your central nervous system and going out towards your peripheral nervous system and going towards your muscles. The other misconception out there is this idea that neurons can somehow give types of information, and that's not really true either. They simply can send a signal, one type of signal. And so they sometimes refer to this concept as the all or nothing concept. You either send a signal or you don't. There is no half signals, quarter signals, nothing like that. And this is a little bit confusing at first because everybody knows you can move your muscles slowly or you can move them fast, and you can also move your muscles with a little bit of force or a lot of force. And so that is where it gets a little confusing. And what that has to do with is not the type of signal that's being received by the muscle, but the number of signals, because there are multiple neurons all going to the same muscle. So the number of signals coming from different neurons and the frequency with which that muscle is receiving that signal. So that's where we are able to control different stages of a muscle movement. It's not necessarily, there's different levels of a muscle movement, fast and strong and things like that. But the individual signals that are being sent by a motor neuron are all the same, and they either happen or they don't. So, and then we talked about this. The signal can only be propagated in one direction, and for the case of motor neurons, they're starting in the spine and working their way out to the periphery of your body. Here is a neuron. Now, I want to clarify this at first because a lot of people assume this is exactly what all neurons look like, and that's not true. But this is a general idea of what a neuron looks like. And it's unfortunately not to scale because this long skinny part is going to actually be much, much longer in most cases. So for example, you might have a neuron that this part of the neuron is in the base of your spine, whereas the other end of your neuron is at the end of your big toe. And that is one cell that goes all that distance. So obviously this in-between part, this skinny part, is going to be much, much longer. And the degree of branching really varies from cell to cell. So that's another difficult thing. But here's the general anatomy. Up here, we have the part of the cell that is going to send the signal. Okay? Now it has several important parts. One is the nucleus, of course. Every cell needs that to be able to make the proteins and carry the DNA. It has also these things called dendrites. Now, the dendrites are really reaching out and receiving signals from other things. Dendrites are able to collect information from other neurons or possibly other things. So, for example, this not a motor neuron, but a sensory neuron. Out on these dendrites, you might have sensors for certain chemicals that might be on your taste buds or in your nose. Or you might have sensors for pressure that might be on the tips of your finger. And those are going to pick up the signal that will eventually get sent all the way down this neuron. But in the case of motor neurons, things that are being sent out, this is usually attached to other nerves, other neurons. Okay, they're receiving, and eventually we will get to that point where that signal is being collected at something called the axon hillock. That's just sort of the neck of the body. So we have the body of the neuron, and then we have the axon hillock, which is essentially the neck. And then we have this long skinny piece. Okay, that entire long skinny piece is called the axon. And again, they can be quite long. The axon is almost completely covered in these specialized cells called Schwann cells. Schwann cells are basically fat cells. They have very little else in them, and they're extremely important, but they do very little by themselves. But they are an important accessory cell to this neuron. So we have the axon, the whole long skinny part, covered by Schwann cells, and collectively speaking, the Schwann cells are called what is known as the myelin sheath, and they're a protective layer. Think a little bit like a wire that's running through your house that's covered in that rubber, the rubber on the outside, to make sure that the current that is going through there doesn't cross over and spark into another wire. That's sort of the idea here, and we'll get more into the details of exactly what these are doing. And then down near the bottom, we branch off into these little terminals, and those little terminals at the end are called presynaptic terminals. I think one thing I missed is this nodes of Ranvier, these little spaces in between the Schwann cells. So they're going to be significant later on as well. Okay. All right, membrane potential. So one of the key features of all nerves is that they send electrical signals, and electrical signals have to be created. You cannot just send a zap down them. In fact, what is the case with nerves is they actually create a charge and then sort of release that charge when they are ready to fire. And these charges are very minuscule. They're measured in millivolts, not voltage, so it's not like you can feel them, but it is why your body can be affected by electric stimulus. So anybody that's used like a stim machine, or I think it's something that's called TENS or something like that are actually make your muscles actually contract because they're actually putting the charge in there. They're replacing what your neurons would do and getting those signals to the muscles. So two types of charges that we'll talk about. One is called the resting potential. Resting potential is the state of a nerve when it is inactive. Now, the one misconception out there is resting potential sounds like it does not require energy. And in fact, creating the resting potential does require energy. But the neuron itself is not firing, which is why it gets its name resting potential. So you're actually going to work, your nerves are working 24 hours a day, 7 days a week to be ready to fire. And to be ready to fire, you have to establish this resting potential. The action potential is the changes that occur when the signal is actually being sent. And so that is going to be the release of that energy. So this is a really important graph that we're going to talk about a little bit more. So this is what this was done was actually using a voltage machine hooked up to neurons. And what's happening here is what you see is a nerve signal that zooms by a particular location in a neuron, somewhere along the axon. So what we see here, down here, this negative 90 millivolts is actually resting potential. That is what a nerve is at when it is not firing. And it takes some energy to make sure that that happens. But what happens is you'll see that you'll start to get some changes, and we'll get to this imaginary line called a threshold where the neuron will actually fire. And it creates a very rapid, what is called depolarization, which essentially is the release of that energy that is stored up by the cell, and then a rapid repolarization to the point where you actually go over and you get to what is called a little step called hyperpolarization, and then you get back to resting. All of this takes a couple milliseconds. So establishing resting potential, as I said, the name is as a misconception because you actually need to work constantly to do that. So you establish it by something called the sodium-potassium pump. Sodium-potassium pump is a very common pump in lots of cells, but particularly in neurons. And its job is to pump sodium and potassium. It does this in two directions. It actually pumps sodium outside of the cell and pumps potassium into the cell. And it does it, it uses, for every cycle, it uses one ATP molecule. And while it does that, it can actually pump 3 sodium out and pump 2 potassium in. It is a very, very common pump, as I said, in virtually all cells, but it is really, really important in this neuron. So this is a video, and let me see if it'll work on here. So I think the sound is turned down here, but I'll talk yourselves through it. So this is the outside of the cell, the inside of the cell down here. There's our ATP. Our sodium gets pumped out of the cell. And then, as these sodiums are released, potassium is going to come in from the outside of the cell. And when we get there, it's going to pump that potassium back in. And what's going to happen is that is going to create a situation where you have lots of sodium out here and very little sodium on the inside, and lots of potassium on the inside and very little potassium on the outside. So they're going to show you one more cycle real quick. So ATP comes in, gives its energy, pumps sodium out, and then we pump potassium in. Okay, and it only requires one ATP molecule per cycle. Okay. All right. And one last concept here is this difference between ion channels and ion pumps. Channels allow things to flow down the concentration, so they do not need energy. They also have gates on them, some of them, that can be opened and closed by different things. So ion channels are passive, do not require energy, and they just let things flow across the membrane. Ion pumps, and the one that's the most important is that one we just looked at, is the sodium-potassium pump things against their concentration gradient, and they require ATP to do work. This is what our setup is along our cell membrane to establish this resting potential. So we have lots of sodium on the outside, lots of potassium on the inside that's established by that sodium-potassium pump. And then we have these ion channels, and you can see that there's both sodium and potassium channels. Potassium's going to want to go out, sodium's going to want to come in based on their concentration gradients. But right now, all of these are closed. And that is the resting state of neurons. We'll get to more the next time. Thanks, guys.